Today we are going to start the second chapter of PATHOMA, and our topic for today is inflammation. We'll discuss some important points of acute inflammation followed by the mediators of the acute inflammation. So let's get started. First of all, the definition of inflammation is that the body's response to a pathogen or an antigen in which the inflammatory cells, the plasma proteins, and the fluid exit from the blood vessels and enter the interstitial space, okay, in which all the contents from the blood are exiting from that blood vessel and entering into the tissue space is called as the inflammation. This is like the basic concept of the inflammation, either it is the acute or chronic, okay. First, we are discussing about the acute inflammation. There are two hallmarks for the acute inflammation. Hallmark means that the two important things that you are going to see in the acute inflammation. First is the edema, okay? And edema is basically due to the fluid leakage from the capillaries into the interstitium. And the second important inflammatory cell is the neutrophils. Neutrophils are white blood cells, and these are very important in the acute inflammation. Contrast to the chronic inflammation, in which we are going to see the lymphocytes. The two important stimuli for the acute inflammation is first is the infection, either the bacterial or viral infection, and the second is the tissue necrosis. Tissue necrosis, as you know, that in contrast to apoptosis, apoptosis does not lead to inflammation, but necrosis always leads to inflammation. And the second stimuli for inflammation is the necrosis. What are the goals of the inflammation in case of infection and necrosis? In case of infection, the inflammation's goal is to eliminate that pathogen, and in case of necrosis, the goal is to clear the necrotic debris, okay. We need to clear that debris which is present in the necrotic area. Other important properties for acute inflammation, that is it is an immediate response, okay, and it has limited specificity. It means that it is a generalized response, it is not a specific response, in contrast to chronic inflammation that has lymphocytes, and lymphocytes has B and T types. The B lymphocytes produce specific antibodies, immunoglobulins, against the antigens, but in case of acute inflammation, the response is not specific. It's like more towards the generalized response, and it is a component of innate immunity. What is innate immunity? Innate immunity is the body's first line defense, for example, over skin, over mucous membranes, okay? And innate immunity is also rapid and non-specific, like defense mechanism against the pathogens, in contrast to adaptive immunity, in which the immunity is specific due to the production of antibodies by the B lymphocytes. So what is to take home? Acute inflammation is characterized by edema and neutrophils. The stimuli are infection and necrosis. It's a type of innate immunity. It is an immediate response with limited specificity, okay. In this histological specimen, you can see that these purple bilobulated cells are neutrophils. Okay, these are neutrophils present in the interstitium. These are the two blood vessels, and we are mostly focusing on the tissue space. And these like spaces present in between is the edematic fluid. It is the fluid which we call as exudate. For example, myocardial infarction. What is myocardial infarction? It is a type of necrosis, right? And 24 hours after the infarction occurs, there is acute inflammatory response. And in that patient's body, what we are going to see? We are going to see the neutrophils. As we have said, that in case of acute inflammation, hallmark, the white blood cells are the neutrophils. Let's talk about the mediators of the acute inflammation. These are very important and very high yield, and there are a lot of them, and we will discuss them one by one. So first is the toll-like receptors. Toll-like receptors are the mediators of acute inflammation. What is mediator? Mediator is something that mediates or that causes or that promotes the inflammation, okay? Toll-like receptors are present on the cells of the innate immune system. It is present on the macrophages and the dendritic cells. What is the function of TLR? TLR function is to identify the pathogen, right? And how it will going to identify the pathogen? The pathogens have PAMPs. PAMPs is known as the pathogen associated molecular patterns, and these are recognized by the TLRs. Okay, toll-like receptor recognizes these PAMPs and then mediate the inflammation. Let us see how it's going to mediate the inflammation. Let's see step by step. CD14, okay, CD14 is a toll-like receptor that is present on the cell of innate immunity known as macrophages. It recognizes the LPS. What is LPS? LPS is lipopolysaccharide. It is present on the outer surface of gram-negative bacteria. And what is LPS? It is a PAMP, right? Because PAMP is present on microbes. So CD14 recognizes the LPS, and what it's going to do? It causes the activation of toll-like receptor. It leads to the activation of CD14, i.e. TLR. It's going to upregulate the nuclear transcription factor kappa B, NFKB. These are very important points to remember, that nuclear transcription factor kappa B is going to upregulate due to the activation of the toll-like receptors by the recognition of PAMPs. Okay. After that, nuclear transcription factor kappa B activates the immune response genes. It causes the activation of genes, and these genes produce the immune mediators. Okay, understand that how toll-like receptor mediate inflammation. They recognizes PAMP. PAMP leads to the activation of toll-like receptors. They causes the upregulation of NF-kappa B, and they activate the genes and then the production of immune mediators. Toll-like receptors are not only present on the innate immunity cells but also present on adaptive immunity cells, i.e. lymphocytes. The second important mediator is arachidonic acid. Arachidonic acid, from where it is produced? It is produced by the phospholipid cell membrane by the enzyme phospholipase A2. Okay, arachidonic acid is produced by the phospholipase A2 from the phospholipid cell membranes, and this arachidonic acid AA, okay, is going to be acted upon by two pathways. It will going to undergo either one of the two pathways. First is the cyclooxygenase pathway, and second is 5-lipooxygenase pathway, and these pathways will produce their own specific products. In case of cyclooxygenase pathway, the product is going to produce is the prostaglandins, and in case of 5-lipooxygenase pathway, the product is going to be leukotrienes. Okay, what are the types of prostaglandins as far as the cyclooxygenase pathway is concerned? The prostaglandins are I2, D2, and E2. These are also very important mediators of acute inflammation. They mediate vasodilation. They causes the increased vascular permeability. Why vasodilation, increased vascular permeability is important? Because inflammation is the leakage of fluid and blood content from the blood capillary into the interstitium, so that vasodilation and increased vascular permeability plays a role in case of inflammation, right? It's important. Also, prostaglandin E2 mediates pain and fever. So these functions are mentioned. One of the most important thing mentioned worth is that vasodilation occurs at the level of arteriole. Okay, and increased vascular permeability is going to occur at the level of post capillary venue. These two like locations for these two specific functions is very important. Second pathway to which the arachidonic acid is going to is the 5-lipoxygenase pathway. It's going to produce leukotrienes. What are the types of leukotrienes? Leukotriene B4, C4, D4, and E4. B4, what is the function of B4? Has a very specific function. It is a chemotactic for neutrophils. It attracts and activates neutrophils. As neutrophils are hallmark for acute inflammation, so these need to be activated by some means. So leukotriene B4 is one of them. Other activators or mediators or like chemotactic substances for neutrophils are C5a, it's a complement protein, interleukin 8 and bacterial products. These four are the chemotactic for neutrophils. First, leukotriene B4, C5a, interleukin 8 and bacterial products. Other leukotrienes C4, D4 and E4 also causes vasoconstriction, bronchospasm and increased vascular permeability. But how? Okay. Important thing here to remember is that C4, D4 and E4 causes smooth muscle contraction. Okay. When smooth muscles are contracted in the arteriole, they causes vasoconstriction, right? When smooth muscles are contracted in the bronchus, it causes bronchospasm. And when the smooth muscles in the arteriole, in the pericyte contract, they increases the vascular permeability. Let me show you how. These are the endothelial cells of the capillary, right? And in between these endothelial cells are the pericytes. These are like pericytes. The pericytes also have contractile function. And when these, for example, this pericyte is contracted, the space between these two cells is going to increase. So increase vascular permeability. Okay, like these are the substances of anaphylaxis. Anaphylaxis is a condition, it's a type 1 hypersensitivity in which there is bronchospasm, vasoconstriction and increased vascular permeability. So these three are also reacting substances of anaphylaxis. Okay, we have discussed two mediators. First, to like receptors, second arachidonic acid, and then mast cells. Mast cells are widely distributed throughout the connective tissue spaces, okay, and these are activated by which things? The mast cells are activated by tissue trauma, okay, if there is trauma to the tissue, it will going to activate the mast cells. Second, complement protein C3a and C5a. We'll discuss them in a minute, but these two, remember, 3A and C5A will going to activate the mast cells. And the third is the cross-linking of cell surface IgE by antigen. What does it mean? It means that this is a mast cell, okay, it's a mast cell. It has multiple IgE immunoglobulin E antibodies on its surface. They are already bound to the mast cells, okay. But when an antigen comes, it cross-link these two IgE molecules, or it can cross-link multiple IgE antibodies, right? IgE molecules or IgE antibodies. When this cross-linking of surface IgE antibodies of the mast cell occur, it will going to activate the mast cell due to the antigen cross-linking the IgE molecules. After the activation of mast cells, what are the like responses or what are the functions of the mast cell? There are two types of responses. First is the immediate and second is the delayed. Immediate response of the mast cell, they releases the preformed histamine granules, okay, they releases the histamine granules. And what is the function of histamine? It's very important. Two major functions. First, vasodilation of the arterioles and increased vascular permeability at the level of what? At the level of post capillary venule, as I have shown you that vasodilation occurs at the level of arteriole and vascular permeability occurs at the level of post capillary venule. So these both are the function of histamine. Delayed response of mast cell is the production of arachidonic acid metabolites. What are the two products of arachidonic acid metabolites? First is the prostaglandins and second is the leukotrienes. But in case of delayed response, leukotrienes are going to be produced. It's very important. Let's see like question or clinical vignette that, for example, if a patient has acute inflammatory response several hours later, acute inflammation continues. What is the mechanism by which mast cells allows the progression of acute inflammatory response? It's the delayed response of mast cells. It is due to the production of arachidonic acid metabolites, i.e. leukotrienes. Leukotrienes maintain the inflammatory response after several hours. The fourth mediator of acute inflammation is the complement proteins, as I have discussed the C3A, C5A, they activate the mast cells, right? So complement proteins, these are pro-inflammatory serum proteins that assist inflammation, okay? These are present in inactive form, but they can be activated. How they are activated, how these complement proteins are activated, they are activated by one of the three pathways. First is the classical pathway, second is alternative, third is the mannose binding lectin pathway. Classical pathway, C1 binds to IgG or IgM that is already bound to the antigen. For example, this is IgG, it is bound to the antigen, right? When C1 binds to the complex of the antigen antibody, it's going to activate the complement system. Okay, it's the classical pathway by which complement proteins are going to be activated. Second is the alternative, the microbial products. Okay, the products present on the microbes, for example, lipopolysaccharides, as present on the gram-negative bacteria, they're going to activate the complement. It's very simple. And the third is the mannose binding lectin pathway. Mannose binding lectin pathway binds to the mannose, okay, and mannose is something that is present on the microorganism. When MBL, okay, mannose binding lectin, it's a molecule. When it's binding to the mannose that is present on the microorganism, it will going to activate the complement proteins. And these three ways by which complement proteins are going to be activated, and they have their own functions. Let's discuss about the result of these pathways. Either the classical pathway is running or alternative or the mannose binding lectin pathway. These all pathways have the same result, okay? They are going to produce C3 convertase. Okay, they are going to produce C3 convertase, and what C3 convertase is going to do, it is going to convert C3 to C3A and C3B. These all are complement proteins. Okay, these all pathways are going to produce C3 convertase. C3 convertase will convert C3 to C3A and C3B. C3b is going to form C5 convertase. Okay, C5 convertase will convert C5 to C5a and C5b. C5b is going to combine with C6 to C9. It means that C6, C7, C8 and C9, it will going to combine with C5b and form membrane attack complex. And this membrane attack complex produces a hole in the cell membrane of the microbe and it's going to attack it. Let's revise it. C3 convertase is going to be formed, converts C3 to C3A, C3B. C3B forms C5 convertase. It converts C5 to C5A, C5B. C5B combines with C6 to C9 and forms MAC, i.e. membrane attack complex. What are the functions of these complements? C3 and C5A. What I have told you, they causes mast cell degranulation. It means that histamine is going to be released. And what is the function of histamine? Vasodilation and vascular permeability increased. C5A is a chemotactic for neutrophils. What are three other molecules or chemotactic for neutrophils? It's IL-8, interleukin-8, okay, these are bacterial products, and C5A, as we have discussed here, C5A, IL-8, bacterial products, and leukotriene B4. These four are the chemotactic for the neutrophils. What is the function of C3b? C3b occurs as opsonin for phagocytosis. As you know that macrophages' function is to phagocytose the foreign material. But how they are going to identify that this material or this microbe or this bacteria is going to be phagocytosed? It's like tagging the foreign microorganism. Opsonin is like the tag proteins. They are going to tag the foreign microorganism, and this is the signal to the macrophages to ingest that microorganism. It's like a tag. What is the function of membrane attack complex? They are going to create a hole in the cell membrane and they are going to lyse the microbes. Of the acute inflammation is the Hageman factor. It is also an inactive pro-inflammatory protein. It is produced in the liver. And how the Hageman factor is going to be activated? It activates upon exposure to the subendothelial or tissue collagen. Okay, it's going to activate upon exposure to subendothelial or tissue collagen, and this Hageman factor is going to activate three systems. First is the coagulation and fibrinolytic system, second is the complement system, and third is the kinin system. This Hageman factor activates coagulation and fibrinolytic system, it activates complement system, and it activates the kinin system. Kinin system has some explanation that kinin is a molecule that cleaves the high molecular weight kininogen. It's like a precursor. It is converted to bradykinin, and this bradykinin is a mediator. It mediates vasodilation and increased vascular permeability like histamine. It has similar function to histamine as well as the pain. Bradykinin also is involved in the pain. What are other mediators involved in the pain? The prostaglandin E2 and bradykinin. These are most important mediators of the pain. So this was the Hageman factor. Let's discuss about the cardinal signs of inflammation last but not the least. There are several cardinal signs of inflammation. They are going to show us that inflammation is going on. First is the redness and warmth. Redness is called in the Latin rubor, warmth is calor. Why the redness and warmth is going to occur at the place of inflammation? Due to vasodilation, the blood flow will going to increase, right? So blood is red and blood is warm. That's why we are going to see redness and warmth. Due to relaxation of arterial smooth muscle as the function of histamine and prostaglandins and bradykinin, they cause vasodilation and increase vascular permeability, right? So they causes redness and warmth at the area of inflammation. Second is the swelling. It's tumor. Tumor is going to be seen due to the leakage of fluid from the post-capillary venule into the tissue space, and it's called as the exudate in simple words. Where is the vascular permeability is going to increase? It is increased at the level of post-capillary venules, okay? And fluid is leaked into the tissue space. We call it as the exudate, and that's why swelling is there. And mediators are histamine because it causes endothelial cell contraction. And when endothelial cells are contracted, the space between them dilates, and the tissue and the fluid passes through that space into the tissue space. Second is the tissue damage. For example, endothelial cell disruption occurs and the fluid can easily pass through the blood vessel into the tissue space. So these both are mediators of the swelling. Pain. Pain is due to the bradykinin and prostaglandin E2, and how the pain is going to occur, the physiology behind is that the sensitization of the sensory nerve endings. It's going to develop the pain. And the last is the fever. Fever is going to occur due to the pyrogens. What are pyrogens? Pyrogens are the products from the bacteria or like fever causing agents. In this case, example is the lipopolysaccharides from the bacteria. They cause the macrophage to release interleukin 1 and tumor necrosis factor. Okay. These interleukin-1 and tumor necrosis factor increases the cyclooxygenase activity in the perivascular cells of the hypothalamus. What is the function of cyclooxygenase? Cyclooxygenase has the function to convert arachidonic acid to the prostaglandins, right? We have discussed here that arachidonic acid, when undergoes the cyclooxygenase pathway, is going to produce prostaglandins. Okay, so cyclooxygenase is an enzyme that converts arachidonic acid to prostaglandins in the perivascular cells of the hypothalamus, and this prostaglandin E2 is going to raise the temperature set point of the hypothalamus, and that's the fever. Then we are going to experience the fever, and these all are the hallmark and cardinal signs of the inflammation. This was for today, and in the next video, we'll discuss how the inflammation is going to occur, the chemotaxis, the immigration, and all of that stuff. So, take care, and till then, goodbye.